Dr. James A. Shapiro, welcome.
Thank you.
Thanks for making the time for us.
Well, it's a pleasure.
So a little introduction about you. You've got a bachelor's from Harvard and an associate's from Cambridge. You've been the professor in the Department of Biochemistry and Molecular Biology at the University of Chicago since 1973.
Yes.
And you won quite a few awards. I didn't write them all down, but
Yes. Well, I've gotten some recognition.
so yeah, I wanted to talk to you about human evolution today. And we spoke for a second just before this, but the one thing I wanted to cover first before we get into your work is the evolution of humans, that we came from primates. And the more I think about it, the more I wonder why we don't see primates continuing to evolve, why there aren't the steps between primates and humans that exist on the earth all the time, why it was a one-time thing and it hasn't happened again since.
Well, we know that there have been different forms of primates that resemble humans. They're called hominids, and they appear, and then all of a sudden you have one type for a period of time, and then it disappears, and a new type appears. That's called punctuated equilibrium. And that's been a process which has been going on. Humans, as we know them, are only about 100,000 years old or so. And that's not a very long time in evolution.
Yeah.
So the chances that we're going to see a major change uh, are slight, except, of course, now we're going into climate change. That's going to put a lot of pressure on living organisms to adapt. And we may see an increase in evolutionary changes.
What are your feelings about climate change? Do you think it's caused by us, or do you think it's just the natural order of the world and it's happened dozens of times before us and it'll continue to happen whether we do anything about it or not?
Well, I think we're responsible for a lot of it because we've been so successful and changed the cover topology of the Earth so much uh, that and the atmosphere, of course, uh, that we've taken away the natural cooling systems which had evolved, basically forests, and so I think we were responsible for it. And we should stop worrying about uh, profits for the oil and gas industries
Yeah.
and get busy doing something.
Yeah. Alternative energy.
Exactly.
Yeah.
And
Hydrogen or
yeah, and alternative ways that we live our lives so that we create less pollution and use less energy.
yeah. I mean, they've got the big electronic car boom going on right now, but from what I understand, it makes more pollution creating those cars than it saves with them being on the road.
That could be the case.
Yeah. Might not be the answer in the long run.
Well, we'll have to see.
Yep. Time will tell.
Public transportation is important, but we don't put the resources into it we should.
Yep. I agree. So you've written quite a bit of papers, and you were the lead author on the first team to isolate a single gene from an organism. Can you tell me a little bit about that?
Well, that was at the beginning of my career,
Yeah, back in your
and
college.
well, no, I already had my PhD when we did that work, but
Okay.
um, it just we were playing around with incorporating parts of the E. coli genome into viruses, and using them to transfer those parts of the genome from one organism to another. And it appeared to me that it would be possible to isolate a pure segment of the genome by taking the viral DNA, where uh, those segments are enriched by about 100 to 1,000 fold, and hybridizing them and chewing away the rest of the unhybridized DNA. And that happened to work. And so we published that.
Wow.
I don't like to use the term gene actually, because a gene has a lot of connotations which I think are probably incorrect. Uh, it was called George Beadle called the gene the basic unit of life, but genes really aren't units because they've got all different parts. They've got signals for transcription, signals for splicing, signals encoding parts of the protein, and so forth. And they're not unitary. Each genetic locus, which encodes a protein or several proteins, is a system and needs to be treated that way. We had the idea that back in the 1940s and up through the 60s and 70s that you could treat things as fixed units, but they're really changeable and they're complicated systems, they're not units. So I always use the term genetic locus.
Okay. What about natural genetic engineering?
Well, uh, that's central to my view of evolution. And if you just remember me to pull up some notes here.
Sure.
Yeah, so all living organisms have the biological tools to change their genomes when that becomes necessary. Our ability to isolate and sequence DNA has helped us learn a great deal about the processes involved in genome evolution. The results are very different from what people are taught in biology classes in schools and even to graduate students in universities. We now know about many biochemical tools that living cells possess to rearrange and rewrite their genomes when they are challenged so that normal reproduction becomes difficult. Genomes do not change by random mutations and gradual natural selection through survival of the fittest. They change by merging cells, acquiring DNA from other organisms, viral infections, and rearranging their own genomes in several different ways. I like to call many of these biological processes natural genetic engineering because they involve the cutting and splicing of DNA molecules, just as we do in biotechnology. You should keep in mind that when we use sophisticated genetic engineering tools like CRISPRs, we didn't invent them, we got them from bacteria and archaea, basic cells without a nuclear membrane that had been evolving for 3.5 billion years. Does that make sense to you?
Yeah. Yeah, absolutely.
Yeah. And that's
Um,
how we can go through some of those steps in evolution that people don't think about generally.
yeah, that sounds great.
Yeah. So the first step is symbiogenesis, which means uh, living together to create something new. And that's when cells fuse in evolution. And we can see examples of that in the fact that uh, all higher organism cells, eukaryotes, use oxygen and grow aerobically because they have mitochondria. They can use the oxygen for metabolism. Now, bacteria and archaea have been around for 3.5 billion years or more. Eukaryotes have only been around for less than 2 billion years. And we now know from sequencing the DNA in the mitochondria that it was once a bacterium. Uh, um, and uh, the bacterium fused with an archaea to create a cell which eventually ended up being a eukaryote. We don't know the full history of that evolution, but we do know that my ancestor of the mitochondrion was an endosymbiotic uh, bacterium living inside another cell. And another way to think about this is when we look at photosynthesis in eukaryotes. Um, Algae, plants, and so forth. They all have what are called chloroplasts or plastids, and those come from endosymbiotic cyanobacteria. Without them, plants would not have photosynthesis. And again, that's established through sequencing the DNA in the chloroplasts to show that they are related to the cyanobacteria. And there are bacteria endosymbionts going on today in aphids and many other insects. And we ourselves have endosymbionts. We call them the gut microbiome. And that affects many features of our lives. The bacteria that we have in our intestine affects how we behave, how we feel, what our mood is, because these bacteria are essential for synthesizing neurotransmitter peptides. So organisms are, again, like genes, they're not simple unitary objects. They are composites put together in the course of evolution.
Wow.
Another thing that is come out of the DNA sequencing is that organisms don't just get their genomes from their parents. There's what we call horizontal transfer across species boundaries. And we see this in a practical way in evolution of antibiotic resistance in bacteria, because we're now finding it difficult to use our antibiotics because we get multiple resistant superbugs, and they're untreatable and they cause untreatable disease. And this is against the established conventional view of how antibiotic resistance arose by mutation. But we now know that these bacteria have plasmids which can transfer resistances from cell to cell. And on these plasmids are structures which can incorporate DNA encoding new resistances. So you can go from a transmissible genetic element from one cell to another, which can give resistance to one antibiotic, say penicillins, and then it acquires resistances by integrating other DNA to give resistance to other antibiotics, like the streptomycin and the related antibiotics there. So the ability of these cells to transfer DNA is very important, and it's very widespread in bacteria and archaea, which are organisms which don't have a distinct nucleus. They can transfer DNA. We find that some of the ways that bacteria become pathogenic is by acquiring the ability to produce toxins. And in many cases, these toxins are carried by viruses. And the viruses incorporate into the genome of the bacteria, which then makes the toxin. And so these are insertions by a special mechanism called sequence-specific recombination. And that was something that wasn't known until about the 1960s. So acquiring characteristics from viruses, and there are toxins in plant pathogenic fungi, and they're acquired sometimes from bacteria, sometimes from other fungi. Um, an example of what I'm talking about horizontal transfer is there are certain insects which have evolved, excuse me, to be able to nourish themselves by eating plant material. But the problem with plant material is it's mostly very long, complex polymers which are hard to digest. And in different uh, um, herbivore organisms, basically nematode worms and beetles, but in others as well, it turns out that they acquire the ability to digest this plant material by acquiring DNA from bacteria or from fungi and integrating it into their own DNA. And so that says that the genetic material that encodes these enzymes can be acquired by a cell and it can be integrated into the genome. So this is all part of what I call natural genetic engineering.
So they start eating the plants and their body adapts to it, and then they can eventually break it down and process it.
Well, yeah, it's hard to know what the connection is between the horizontal transfer there of the ability to digest the plant polymers and the acquisition of the traits that make the organism able to feed on the plants. There's a lot we don't understand about the evolutionary process, but we know that they wouldn't have acquired these enzymes from their ancestors because we can identify the ancestors and we can identify the DNA sequences they have, and we see that these uh, plant digestion enzymes are similar to those found in bacteria or in fungi. And that means that the DNA from the bacteria or the fungus has to have entered the cell. That's the whole complex process that we need to understand, which we don't. And then the cell is capable of uh, cloning that DNA into its genome. And that's another example of what I mean by natural genetic engineering.
The two DNAs combine themselves.
Exactly.
Wow.
Yeah, basically it's the acquired characteristic is integrated into the larger genome of the whole organism. I would suspect that probably precedes the evolution to be a specialist on plants because it gives the organism the ability to digest plants, and from there they can evolve to be even better at it.
Very interesting.
Yeah, and we know that proteins don't evolve as we were taught by one amino acid at a time due to random mutations or replication errors. Um, Comparing many proteins, now that we can do genome sequencing, it's clear that the proteins are kind of like have a kind of Lego-like structure. Proteins are composites uh, composed of regions called domains. And you can see the same domain in two or three or four different proteins doing different jobs. But it's then in combination with other domains. And the domains have specific activities like carrying out a chemical reaction or binding to a specific region of DNA or binding to other proteins. Um, and these domains get shuffled, as they say, in the course of evolution. And what that tells us is that the evolution of these proteins occurred by cutting the coding sequences or the genetic loci which encode them and creating new mixtures of exons and introns so that we get the new proteins formed. And
That's something they do naturally, or
yes,
that's
it's something they do naturally.
how similar is that to CRISPR?
Well, CRISPR is quite different, actually. Uh, CRISPR targets specific sequences and cleaves the DNA there. It can do other things as well, but that's the more general form. And CRISPR has its own natural genetic engineering involved in acquiring the sequences which allow it to target, say, an infecting virus. If a cell survives a viral infection, the cell can pick up a piece of the virus sequence and integrate it into its CRISPR system. And that allows the cell to make an RNA which recognizes that sequence and can make it easier for the cell to knock out the virus on the next infection.
That's
And
similar to like the RNA COVID vaccine. Is that the same kind of thing that does?
well, um, no, in this case, the RNA plays a targeting role. In the COVID vaccine, the RNA encodes a protein, which is uh, similar in recognition by the immune system to the COVID virus. And that's how that works.
Gotcha.
They chose the spike protein of the COVID virus. And I've begun wondering whether that was the best choice to make because we had so much trouble
Yeah.
with with the COVID switching its spike proteins.
Yeah, lots of vaccines, lots of boosters.
Right. I've had 7 so far.
Yeah, so is my
Uh,
mom.
and I get my A sooner. We could have chosen perhaps other uh, proteins from the virus to choose as antibody targets. But one of the most astonishing capabilities that cells have, and this is true of all cells, both prokaryotes and eukaryotes, is they have what are called transposable repetitive elements. These are pieces of DNA which can move from one place in the genome to another. And there's 30 times more DNA encoding these repetitive elements in our genome than encoding protein. So they're very important. Barbara McClintock, who later won a Nobel Prize, discovered these elements in the 1940s. She called them controlling elements because when they insert, they change the regulation of a genetic locus. And this is part of what I mean by a genetic locus is a system that can bring in new regulatory sequences and change the way a particular locus is expressed in the course of development. And these transposable elements are found in bacteria and all other organisms. They use sequence-specific proteins to move from one location and insert into another in the genome. That's what transpose means. It goes from one place to another. There are different molecular mechanisms for these transposons. There's some that transpose purely as DNA. Some that are called retrotransposons, which have an RNA intermediate, which then gets reverse transcribed into DNA. And uh, uh, Linnaeus, who was the father of taxonomy, used to say nature doesn't make leaps, doesn't make jumps. And Darwin quoted that, I think, 6 times in Origin of Species. But genomes actually do this. They do this routinely and in more than a single way. So uh, that tells us that the ability of cells to manipulate their genomes goes far beyond uh, simple mutations or even chromosome breaks and rejoinings. And these elements can be activated by stress because they're biological, they're regulated, and that links the experience to genome change. So if an organism goes through a tough time, its transposable elements can be activated, and then it will change when they insert it in new locations. And one thing specifically about these changes is that if you have a piece of DNA which will change regulation of a genetic locus, and it can go to multiple different places in the genome, you can create genomic networks by having the transposons go to sites near genetic loci that need to be co-regulated for complex traits. And examples of how this happens are the vertebrate body plan, nervous system development, and pregnancy, where multiple different functions have to coordinate to allow the organisms to proliferate. And
Yeah.
the fact that this DNA, this is what Dawkins called selfish DNA. And people call it selfish DNA because they didn't understand why this DNA was so abundant in genomes. But we now know that the more abundant, this DNA is more abundant in more complex organisms. And this was the so-called C-value paradox, which is the amount of DNA which was in these elements was so much greater than the DNA which was in the protein coding DNA, which was thought to be the important part of the genome. But now we know that genomes encode more than just proteins. They encode RNA molecules, which not only can be messengers to direct the synthesis of proteins, but can also regulate the genome. And there are many more of these regulatory RNAs than there are of proteins. And and these RNAs are encoded generally by DNA, which includes some of these repetitive mobile transposable element DNAs. And just trying to look at my notes here.
Yeah, no problem.
Yeah, it's saying the repetitive DNA actually encodes major parts of the many recently discovered RNAs, or called ncRNAs for non-coding, that play major roles in regulating and executing genome functions in complex development. So that's another discovery that has been made. And that, again, is not consistent with the random mutation natural selection idea of evolution. So is the way a mobile element can help create a network clear for you?
Yeah. So
Okay, good.
you guys have kind of proven a lot of what Darwin had wrong through more
Well,
research.
yeah. Uh, and the thing is that the more we look, and I've got a couple other things to cover yet. The more that we look, the more we see the simple random mutation of small effects, natural selection idea just doesn't explain things.
Mhm.
And one of the reasons for that, I think, is that uh, the random mutation idea really only affects single characters, single properties of the organism, a single genetic locus, for example. And much of the organism is composed of multiple components, um, which have to work together as a network. And that's again taking a little bit more complex view of how living organisms function. And that's what the transposable elements allow the organism to bring those different functions together by moving the same transposable element near the genes, genetic loci, I should say, which encode those proteins.
Hmm.
This is also contrary to the central dogma of molecular biology, which was propounded by Francis Crick. And it always seemed to me strange that you would have a dogma in science. And that central dogma was that the job of DNA was to code for proteins through an RNA intermediate. And now we know that much of the genome encodes uh, these regulatory RNAs that play major roles in regulating and executing genome functions.
A lot of people in the past have gotten things wrong.
Well, I was a good friend of Barbara McClintock's for the last 12 years of her life. And what she always used to talk about was the now explanation. And when you think you've got a final explanation for something, like George Beadle calling the gene the basic unit of life, uh, that's only temporary. You never know what you're going to discover in the future.
Yeah.
So you should always be ready to change your ideas based on what's found later on.
Yeah, have an open mind about it.
Exactly. But it's been difficult to get people to change the way they think about evolution.
Yeah, the mainstream does not like change.
No,
They think they got it right the first time and they wrote books about it and they don't want anyone arguing with it.
right. Yeah.
Yeah, you see it all over the world with everything.
Yes, that's human nature, I guess.
Seems to be. Yep. People need to have a more open mind about things. The more advanced we get, the more technology we have, the more things we'll be discovering.
Right. Yeah. And two more things I'd like to mention.
Sure, go ahead.
Um, one is uh, what's called hybrid speciation. And it turns out that we can actually make species by mating two organisms from different species, but two different related species, which are very similar to each other. And the reason this is important is that if the population of one species declines because of some ecological change, and it's hard for it to find mates of its own species. It will look for the mate that's closest, that most closely resembles the mate it should have, and that will create hybrids. And it turns out that when hybrids are formed, they very often display loss of control of genome expression. They activate their transposable elements. There are changes in chromosome structure. And there's also genome doubling. Uh, the DNA content or chromosome content of the genome just doubles. And these are key steps in forming a new species. This is done most often in plants because you can take the pollen from the plant and fertilize it itself. But even without crosses, uh, the interspecific hybrid with this great genome instability creates species which will have reproductive isolation because they have different chromosome structures and different adaptive characteristics because they have transposable elements moving around in their genome. And they may have other differences as well. I don't think the process has been studied quite as exhaustively as it can be. And we know that this is going on in nature because frequently genome sequencing shows signs that hybridization has occurred of what's called introgression of DNA sequences from related species. And you combining the DNA of two different species together. And this tells us that speciation can be a very rapid process. And it doesn't take long periods of time, long periods of natural selection. Uh, so that's a counter example, if you will.
Yeah,
Uh,
one of the things I read in your paper was, Darwin was convinced it would take millions of years for things to evolve, and you guys were able to do it in a matter of what, months in a lab?
um, well, you can do it in months or depends about the reproductive time of the organism is, but months would be okay.
Yeah.
Yeah, and that's why you have that pattern of punctuated equilibrium.
As far as species combining, have you heard all this talk about bananas going extinct?
I have.
What do you think the answer to that is? Some kind of genetic combination of a banana and plantain or something similar that'll keep it alive or create a new version of itself? Or
Well, I don't know enough about banana biology to be too specific, but I think the idea of many of these plantations were made by not by crossing banana plants, but by taking tissue samples and cloning them. So all of the plants had the same genetic background, and that makes them more susceptible to spread of a disease. So I think we need to have a greater diversity in the genetic composition of the banana plants.
so lots of different kinds of bananas would take care of that for us.
Yeah, I think so.
Hmm.
And uh, it's inevitable, I think, that if you want an example of evolution in action, this is it. Because whenever you have a pure population of many individuals with the same genetic composition, you frequently find pathogens, which will
Problems.
ultimately are able to parasitize them.
Hmm.
And the last thing that we've discovered uh, which again is contrary to what we would learn in studying Mendelian genetics. Um, is a process known as chromoanagenesis, which means chromosome rebirth. And basically, this is multiple chromosome structural changes that occur in a single cell division. And it's often triggered by chromosome breakage. And this was discovered in cancer genomes in 2011. And at first, one might have thought that it was a property of the abnormal cancer cells. But it was quickly observed that this kind of structural rearrangements of many proteins occurs in plants, animals, and eukaryotic microbes in their germlines as well. So this changes the genome in many ways. And what it does is, one thing it does is reorganize the spatial organization of the genome, which is intimately connected to its regulation and expression. But it turns out that the changes go even further because the way the chromosomes get broken and then put back together involves what's called double-strand break repair or non-homologous end joining. And there are two ways this can happen. And one is by a conventional system called conventional non-homologous end joining. And that doesn't make many changes. That basically just ligates the two pieces of DNA together. But the other process involves connecting the ends of proteins where one of the strands has been chewed away. And it requires DNA replication to join the ends together. And when you look at that, you find that a whole bunch of new sequences begin to appear in the junctions. And it involves a very astonishing enzyme called DNA pol theta, DNA polymerase theta, which is capable of synthesizing DNA from different parts of the genome and connecting them together. So you get a kind of composite there. It's capable of making DNA without any template at all. So that's completely de novo DNA. And
Wow, that's amazing.
these are involved in the junctions of these rearranged chromosomes. Not all of them, but many of them. And the potential for generating novel sequences is very great. And this is perhaps the most extreme example I've seen of an activity which something we never would have thought was possible, but we now know is. And it tells us that organisms can really do very amazing things to their genomes. And what we need to figure out now is how do they do it in ways that it results in such well-adapted organisms.
Is that something you've studied in a lab, or is that something you've seen in nature occur?
This occurs in nature.
Yeah,
Yeah. Uh,
plants or
in plants, more the plants is more experimental because they find that when they try to genetically engineer the plants, that they induce this multi-site rearrangement process. But in us, in humans, many examples of this kind of rearrangement have been discovered in people who have certain diseases uh, uh, which result from non-functioning in parts of chromosomes. The one I've looked at most closely was a woman who had trouble getting pregnant, although she eventually did. But she had 157 different changes in her genome. Uh,
Do they know from what?
no,
Just randomly happened to her or
uh,
something she was exposed to?
well, they don't know. I think in that case, they didn't have access to her parents, so they couldn't tell whether it was occurred in her development or in that of her parents.
Something that was passed down to her?
Yeah,
Wow, that's incredible.
yeah. So I think the ability of cells to rewrite their genomes is quite extraordinary. And that's why I like to talk about the read-write genome. We think of the genome as a kind of computer memory often, and we're thinking of it often as a read-only memory, and it has to change by accident, which was the neo-Darwinian model. But it can be rewritten.
It's got a
Uh,
read-write functionality,
yes.
like a memory card.
Well, the memory card just carries the memory, but it has the ability to change the code. The genome is actually not the uh, blueprint for the organism. It's a database out of which encoding proteins and RNAs out of which the organism can be built. Um, but it has to have certain capabilities and certain sequences. And they can be picked up from other organisms or they can be created de novo by some of these processes. And um, I don't think with all the knowledge we have now, we can say that there's any limit that can be put on what can happen with genome rewriting.
Wow, crazy.
Yeah. So with the banana trees, for example,
Hmm.
maybe they have to mate the two different banana trees and make a hybrid and
Yeah, I
see if
think they created one that tasted the best and they just cloned it and
yeah,
that's all we ever get now.
right.
But it seems like hindsight being 2020, that might have been a mistake, huh?
I think so.
Yeah.
Monoculture is not a good idea.
No, not at all. We'd all be inbred if that happened to humans, huh?
Yes,
Yeah. Not a good thing.
everybody be cousins.
Yeah, exactly.
Yeah.
There's a couple people with some kind of far out theories. I don't know how much you look into any of this stuff, but have you done a lot of research with human DNA?
No,
No?
no, I just read papers about it.
There's people that theorize, I mean, if you believe in aliens, which I don't know, with all the UFO sightings the government's released lately, it's hard not to, but there's people that theorize that our DNA was tampered with at one point.
And why do they say that?
Uh, I don't know all the specifics on it. I'm not an expert in that field, but something about telomere number two being capped, and they think it was edited at one point.
Hmm.
I mean, to the point where, like, we used to have telekinesis powers, and they took it away from us so we couldn't overpower them. It's some far out theories, but the more you listen to it, I don't know. It's very intriguing.
Yeah, well, uh, I think we're seeing such uh, astonishing things happen right now in the laboratory. It's difficult to take what looks like an anomalous genetic uh, system and draw conclusions about it
Yeah.
because our knowledge of what's uh, biologically capable possible has increased so much.
Hmm.
Are there other theories out there that you?
Oh, there's lots of theories out there out there in my line of thinking, but like the pyramids. I know this isn't your wheelhouse, but the pyramids just seem like it would be impossible for us to build today. So how did they do it 5, 10, 20,000 years ago if there wasn't some help from somewhere or something else? I don't know how much of research you've done about Egypt or the pyramids or the Serapeum.
The Serapeum, what's that?
It's like an underground maze of tunnels,
Uh-huh.
and there's these gigantic granite boxes down there. I
Uh-huh.
mean, the box itself weighs like 80 tons, and the lids weigh another 40 tons. And I've called a few granite companies in the area and asked them if money was no object, could they build one? And they said absolutely not. I mean, just like this is a box that was abandoned in one of the hallways. You see how large that thing is?
Mm-hmm.
And the entire inside is cut out perfectly perpendicular, perfect 90-degree corners, and polished to a mirror finish.
Yeah.
And and they're all in alcoves. There's like a map of the Serapeum. I don't have these ready to put up on screen, so I'll just show you. But
Wow.
so it's like a maze of tunnels,
Mm-hmm.
and each one of those little black squares is a box, and it's anywhere from 4 to 6 feet below the floor of the tunnel.
Mm-hmm.
And I mean, we don't have a machine today that could even move something that weight down there. And I had been doing a bunch of research about it, and I always thought it was a ramp that led down to the Serapeum, and they slid the boxes down somehow. But no, this is the entrance to the Serapeum. It's a set of stairs carved out of the bedrock.
Mm-hmm.
So, how in the world did these people 10,000 years ago cut these gigantic blocks of granite from a quarry 500 miles away, move them through a mountain range, get them to the Serapeum that's under the Giza Plateau, move them down these steps, and then place them in the alcoves? And nothing's ever been found inside of any of them.
Mm.
I mean, here's one with a lid, just the lid on top of the box, so you can see how gigantic it is.
Yes.
So, I mean, that's another thing that goes with aliens were here, they helped us, they gave us the technology. I mean, there's gigantic blocks of granite left on the Giza Plateau that have 30-inch circular saw marks cut into them. 10,000 years ago, I didn't know we had circular saws or any type of power to power them. And Egyptologists say they were made with copper chisels and stones,
Mm.
which just, I find that really hard to believe. But that just all goes back to there's a guy, Billy Carson. If you look him up, he's real popular on YouTube. And he's got tons of theories about how the aliens were here and the Book of Enoch and all these old scripts and texts that tell our history and that our DNA was edited and the aliens basically use us as slaves. And I mean, it's a serious rabbit hole you can fall down.
Yeah, it's hard to know how you can verify any of that.
Yeah, no, it's impossible. I mean, it's just a gigantic mystery. But
I thought,
here all these things sit in Egypt, and yeah, if money was no object, we couldn't recreate them today the way they are, which is
yeah.
just mind-blowing. And like the pyramid of Giza's got these 70-ton stones that create the ceiling of the king's chamber, and they're 350 feet up in the air, and they're cut and placed so perfectly together you can't fit a human hair between them.
Yeah, well, I haven't seen the pyramids. I've seen the Machu Picchu.
Yeah.
Yeah,
Yeah, there's pyramids all over the world. I mean, they've been finding
yeah,
them in South America, buried under what they thought were hills tops,
right.
and they turn out to be pyramids that are just overgrown with brush.
Yeah.
But yeah,
So,
it's some crazy stuff. I was just curious if you had ever heard of any of it or the DNA editing stuff. I know that's your specialty, the biology part of everything.
yeah,
But yeah, if you ever have some free time and want to fall down a crazy rabbit hole, we'll look up some of the stuff in Egypt. It will blow your mind.
yeah. And I think the crazy rabbit hole in biology was the idea that evolution can occur gradually, and that we didn't understand that organisms have the ability to change themselves.
Yeah, that is crazy. I mean, I took biology in high school, but that was never one of my majors in college. So I don't know nearly as much as I need to know about the subject, but yeah, just the fact that we evolved from primates and you would think they'd keep evolving and there would be different half monkey, half people walking around the earth right now.
Well, the Denisovans and uh, I'm having a senior moment now. The primitive humans, the
Hominids?
Neanderthals.
Neanderthals, yeah.
Yeah. Well, they were related to us, but they've disappeared. And uh, it could well be that we wiped them out.
So do you think primates will ever begin evolving again? Or do you think that was a one something happened and it was a one-time thing and that's the end of it?
Well, I think if we get to the stage where we learn that primates can evolve, that'll mean that we're in a lot of trouble.
Planet of the Apes?
Yeah. So I would rather remain ignorant on that question than be enlightened.
Well, if it takes as long as it takes, we'll all be gone before that happens, right?
Yeah. Yeah. Well, I concerned about my grandchildren.
Yeah. Yeah. I've got three kids myself, so hopefully nothing crazy happens.
Yes. Nothing more crazy than we've done already.
Yeah, exactly. Yeah. It's a scary
There's,
time.
yeah, there's a very good documentary on climate change if you're interested,
Yeah.
called Regenerating Earth
Okay.
by Don Feldman. And he talks about some of the problems with climate change and how to address it.
So you think that's totally a man-made thing, or you think it's partially our fault and partially just the cycle the Earth goes through every so often?
Oh, well, I think it's largely our fault because of this coincidence with the industrialization and the population explosion and the use of fossil fuels and so forth.
Hmm.
I mean, I think the timing is uh, uh, hard to think is just coincidental.
Yeah.
And I
Just
think the rate of change is very high too.
just a matter of time before we get hit by another meteor and start all over.
Uh, well, hopefully that won't happen while we're here.
Anything else you wanted to cover?
No, I think I've covered most of what I wanted to say.
Okay. Well,
So,
I appreciate your time.
okay, and let me know when you have Are you going to edit this at all?
Yeah, yeah, I'll edit it. I'll send you an email when I get it up.
Okay, great. Thank you.
Thank you, Dr.
And
Shapiro.
good luck with your podcast.
There's 8 million to compete with out there, but I'm going to try.
Good.
I
More
pre
power to you.
Thank you, sir. I appreciate you giving me time today.
Okay.
Thank you.
Bye bye.
Bye bye.